Kristen Atchison here, and we are talking about sensation and perception. This time we're going to be talking about vision. So before we can talk about vision, we have to talk about what we're seeing. And what we're seeing in vision is light. Now, light is actually part, what we can see as light is part of the electromagnetic spectrum. Luckily, this is not a physics class, so we're not going to go into great detail about that. But know that it's this visible light that we see is on that same spectrum as things like X-rays and UV rays and radio waves and microwaves and all these other kinds of waves that we can't see. What we can see is a really small portion of that. Now, other animals can see a greater portion than we can, but this is what we can see with our eyes, a pretty small portion of this spectrum. And within this spectrum that we can see, it's broken apart. How we perceive that and how it's broken apart is in terms of wavelengths. Now, these wavelengths of light are really what we perceive as color. So the longer waves we're going to see as reds and oranges. The shorter waves we're going to see as blues. And we'll talk about that a little bit more when we get into the other theories of light, of vision. But it's important to know that what we're looking at, that rainbow, right, that spectrum of light that we can see really just corresponds to a wavelength. And a wavelength is anything between the two peaks. So if you see here, we have different measurements of these wavelengths, and you notice they're being measured between the two peaks. This is the same way that we measured a wavelength when we talked about hearing and we talked about sound waves. It's the exact same way we're doing it here in vision. So in sound, a wavelength was our pitch. In light, a wavelength is color. So again, also not a physiology class or an anatomy class. So we're not going to go into great detail about the eye, but we do need to have the basic spots of it. We have the light. We can see in this example a candle. That image goes in through our cornea, past our iris and pupil to our lens, that kind of changes that information, that flips that information, and then it puts that information on our retina. The retina is the part of our eye that's on the very, very back of our eye, and that's actually where the photoreceptors, the nerve cells that are specially set up to be able to transduce, remember that word, which is basically kind of change that light information, that electromagnetic signal into a neurochemical signal. And so this is happening starting in the retina. There's a lot of other cells, and your book goes into some more detail, but we're going to kind of keep this simplistic. So the retina is where we have those photoreceptors that we'll talk about. On the retina, there's this part of the retina that's called the fovea. When you look at something, if you are looking at the word fovea on this slide, then you are putting the word fovea on your fovea. When you're focusing on something, that really goes onto that fovea. And this is a really highly centralized part of the eye that has a lot of photoreceptors. Now, the rest of your retina still has photoreceptors. We'll see them in different proportions and different kinds. So while you might be looking straight at fovea, you might see all these other things in your periphery. And that's all those images that are still being put onto your retina. Now, where this information leaves your eye and goes to your brain is through the optic nerve, okay? And the optic nerve is basically just a big old bundle of axons, some other kinds of cells, but primarily it's just transmitting that information to different parts of the brain. Now, because these are axons and not kind of cells in their own right or photoreceptors in their own right, where that information leaves the back of the eye is your blind spot. Because there are no photoreceptors on that part of the retina, then because that's kind of the exit, that's the door, right? So if the door is open, you don't have anything there. It's the same way with the optic nerve, and so that creates our blind spot. So again, this information kind of goes in through the eye, and then will get flipped. So that we're seeing kind of our brain, the information that's going to our brain is reversed. So again, this retina is this yellow piece on the back of the eye. And again, this is where that transduction is happening in those photoreceptors, where we're taking that electromagnetic information and we're transducing it into electrochemical signals for the brain. So again, into those neural signals. And so again, this information comes in, it flips it. And again, if we're looking at something, and in this case, the arrow, that arrow is going to be flipped on our, in our visual field. It's going to be flipped on that fovea. So again, that fovea is that center region of the retina. And this is where we're going to have, where we're focusing on something, this is where that information's going. Now, what's really interesting about the fovea is that it's, again, a highly concentrated area that has more photoreceptors than other parts. And it also has different kinds of photoreceptors. So if you look at the image, all the way at the bottom, kind of where the blue is, at the very, very bottom of that image, there are different shapes. Some of them have kind of cone shapes, pointy shaped cells, and some of them have more rounded shape, rod-shaped cells. And that's because we have two kinds of photoreceptors. We have rods and cones. Our fovea is made up of cones. Cones are really good at color. Cones are really good at highly detailed things. And so our fovea is made up exclusively of cones. Now, further out in our retina is where we get those rods, okay? And we'll have cones as well, but we'll have more rods, a greater proportion of rods out in other parts of the retina. But in that fovea, that center region of the retina, where we're really focusing our attention when we're looking at something, is highly centralized with cones. Again, those are those pointy ones down at the bottom. And so again, there are these two kinds of photoreceptors, the rods and the cones, and both of them transduce light into neural signals. They just do different kinds of light. So rods are really sensitive to lower light levels. So they're what really help you see in the dark. So if you wake up in the middle of the night and you're trying to get your way to the bathroom, it's your rods that are helping you get there. Your rods are really what's helping you see, take kind of the most information out of those low light settings. Now, they don't have great acuity. So in the middle of the night, you might still bump into things. You will not be able to tell things about color. They are not going to be great for those detail-oriented things. You have tons of them, though. You have about 120 million of them. So you have lots and lots of these. These are also really good for motion. So if you're looking, at one thing and you see something move out of the corner of your eye, that's your rods detecting it. And you're going to turn and you're going to selectively attend to something else and put that new thing, that moving thing, or that thing that you thought you saw move, on your fovea, on your cones, which have a lot more information about light, in that they can tell us the differences between colors. So if you look at you're trying to pick out a shirt in the dark, you're trying to pick out socks in the dark, that's how you can end up with those mismatched socks. You can end up with the wrong shirt. You can end up with all these different things because your rods can't see color. They can't really detect those things very well, and cones don't work well in low light settings. So cones, we have a lot of fewer of those. Again, they're mostly located on that fovea. They have this really high density of cones and very little rods. But they, again, are what's helping us with acuity. So again, if you're looking at the word, like on the title of this slide, on the word rods, you can see that very, very clearly. Now, you can still see the other things in the periphery, but they're a lot fuzzier, right? And that's, again, those rods don't have great acuity, but those cones do. So again, we have this fovea has this great high density of cones, which are really, really good in acuity. They're really good in color. But we have way, way more rods, which are going to be really helpful in movement detection, are really going to be helpful in low light areas. Their names are really because of how they're shaped. Okay, so I know a lot of times scientists like to name things confusingly. This is not one of the cases. These cells really look like rods and cones. So here we have schematics on the left, and we have actual pictures of them on the right. And again, you can see the cones have that kind of cone-shaped top, where the rods really just look like a long rod going up. So again, they have these different different shapes to them, which is how they get their names. Now, these are neurons, so they have the same kind of basic structure that other neurons do, except they're specialized neurons. So one end of them isn't meant to be connected to another neuron. One end of them is meant to transduce that information. And so again, we'll still have that nucleus where that cell body, where that decision, that all or none decision is made. And then we'll have a very short axon and then a synaptic terminal that will share that information with the next neuron. So again, these are really named for their shapes. And the easy way to remember which one does color is cone starts with a C, and so does color. And so that's a really nice mnemonic to help you remember that. Okay, so we're glossing over all of these things. If you ever to take a perception class, you will have a whole lecture just on the way the information goes from the eye to the back to the brain. In fact, you might have several lectures on it. In fact, if you're interested, you can look at my online lectures on this for the perception class I taught online. So if you've ever heard that the information on the left side of your body goes into your right side of your brain, and the information on the right side of your body goes into the left side of your brain, we talked about that briefly, kind of a little bit, in our brain chapter, that kind of hemispheric specialization, that lateralization. It is true. In case of vision, however, it's not that your left eye goes into your right brain and your right eye goes into the left side of your brain. What it is the information on your left visual field goes into the right side of the brain. And here that's indicated with the red arrows. And the information on your right visual field goes into the left side of your brain, which is indicated by the blue arrows here. So each eye is getting information from both the left and the right side of the body. And they're taking that information and splitting it, and half goes to one side and half goes to the other. This is really going to be helpful in our binocular vision. When you start to talk about depth perception, super important that we have two eyes. Binocular vision, where we're able to see things as an integrated field because we have two eyes. A lot of that is happening because of this way the brain shares the information from the eyes. So the information goes into the retina, goes out the optic nerve, into the next major place that we'll talk about it goes to is the thalamus, and then it goes to the visual cortex, which is in the occipital lobe. So remember, we talked about if you get hit on the back of the head, the cartoons, they see stars. The reason they do that is because you got hit in your visual cortex. And so it might be firing differently, responding differently because of that trauma. So again, this is kind of the way this information goes in. We do have it split left and right. It's just the stuff that's on the left side of your body versus the stuff that's on the right side of your body. That in that visual field is what's getting transposed. Okay, so that's kind of the basics of how vision works, what light is, how the eye works, how it gets into the brain. So that's kind of the sensation piece of it. Now we're going to move into the perceptual piece of it. So once it gets into the brain, what do we do with it? There's different theories on how this works. So we're going to go through two different color theories, the first of which is called the trichromatic theory. Okay, so trichromatic, really easy if you break it down. Tri means three, so we're talking about three pieces of color, chromatic, right? So it's a theory about three colors is really what this trichromatic theory is. It was the first scientific theory of color vision. So it's also a really great historical theory. And it was proposed by Hermann von Helmholtz in a really, really long time ago in the 1800s. So he, lived from 1821 to 1894. So we're talking about a very old historical theory, but it still has a lot of support and a lot of benefits to this theory, which is why we still talk about it. So again, trichromatic meaning three colors. The trichromatic theory of colors says that there are three types of cones. So remember, cones equal color, and they are sensitive to different wavelengths of light. So remember we said that the long wavelengths are those red lights, the short wavelengths are those blue lights, and those medium wavelengths are kind of the green lights. And what von Helmholtz had says, and others have backed up, is that there's these three kinds of cones that respond, are sensitive to these ranges of color. So we'll see that the red light, again, these longer wavelengths between the two peaks, these are wider, they're longer wavelengths. We see the green light, those medium wavelengths. We've got a special cone that allows us to do that. And then we have a third kind of cone that allows us to look at these shorter wavelengths, these blue lights. And that colors other than these three, because obviously you can see more than three colors, really stimulate a combination of cones. And some of the support that we see for this is things like red-green color blindness, which is one of the most common types of color blindness, where people can't distinguish between those. We see this primarily in genetic males, and so we see that there is this kind of more prevalence in that population than we see in other populations. And here's kind of an example of that. If your trichromatic cones aren't working, this just looks like a series of dots to you. They really are dots that are red and orange, some kind of yellows, some greens, some kind of green blues. So they're activating kind of all of the different colors, all the different cones in your color vision. Now, if you do have functioning cones, you should see a number in there. It should, depending on the clarity of your computer, it probably says 74, but if you see something else, that's okay too. That's going to be kind of top-down processing at work there. But probably 74 is what you're seeing. If you don't see anything at all, either your computer's not working very well, or you may have problems with color blindness. So again, color blindness is one of the big supports we see for this trichromatic theory of color vision. Now, the trichromatic theory is not the only theory we have of color vision. We're going to go through a demonstration next before I introduce the next theory. So what I want you to do is I want you to look at this black dot in the center of the screen. Go ahead and pause your video and look at it for about 30 seconds. Okay, once you've done that, and really, I recommend that you do it. I know 30 seconds, you're like, oh my gosh, this is so boring. Why are we doing it? Just do it. It's really good, okay? So look at this for 30 seconds. Now continue to look at the center of this image. What you should see, if you did this for at least 30 seconds, the longer you do it, the better your response that you'll get, is you'll get an after image where things flip. So we'll see yellow in the blue square, we'll see green in the red square, and vice versa, okay? And this really is explained by our opponent process theory. Opponent process theory says that we have colors that oppose each other. And this is similar to what you learned in art class, right? That these opponent colors, blue and yellow, green and red, black and white, that these are opposing pairs. And what they're saying is that we really have these two sets of color-sensitive neurons. We have red-green and we have blue-yellow. And each color opposes the other. And so because of that, each one can't be stimulated at the same time. Your nervous system is also efficient. And so if it's getting the same input over and over and over and over again, it's going to suggest, hey, we're getting the same information over and over and over again. And so if you're focusing on that black dot in the middle of the four-color square, it's getting that same information of red, green, blue, yellow in those four boxes. And what happens is after that color information goes away and we went to the gray background, those neurons are still kind of firing. And so what you get is those opposite colors. And so again, this is supported, these kinds of after images are supported by this opponent process theory. Again, black and white are also a pair. So if you've ever seen like an American flag that's like green and yellow and black, and you stare at it for a while, and then you look at a white surface, you'll see the red, white, and blue American flag. Same thing, also an opponent process theory after images. So again, the opponent process theory says that we have these these pairs of these neurons that are sensitive to two. And because they can't fire to both blue and yellow at the same time, that we get these kind of overreactions when we go from looking at blue to seeing yellow in an after image, or red to green, black to white, etc., etc. Okay, so that kind of ends us mostly for color. We're going to move on to just visual perception as a whole, how we organize the world around us. So there are so many things in your visual field. There's so many things you see. How does your brain decide when one thing stops and another thing starts? How does your brain decide how far things are away? How does your brain decide what's moving, what's still? How does your brain decide what stays the same and what changes? And this is how we organize and interpret this information that's coming in. And we'll talk about each of these in detail. So we'll talk about how your brain perceives shape. We'll talk about depth perception. We'll talk about motion. And we'll talk about perceptual constancies. So the first one we're going to talk about is shape. And the way that we're mostly going to focus on shape, there's lots and lots of other ways that the brain organizes shape. But one of the ways it does is by grouping things. And the first researchers to really talk about this and develop a really good, stable theory of how we organize shapes in our world really were the Gestalt psychologists. These were psychologists that started their research in Germany. And so Gestalt really means a form or a whole, okay? So it's, again, talking about this shape perception. And it's, again, a theory that talks about how people organize this information, this sensation that's coming in, into a meaningful whole. How do you decide that that's a door and not just a series of little blocks? How do you decide that your device that you're watching on this is separate from everything else in your visual field? How does your brain make that perception? And so again, this is about how they're organized according to certain patterns in our environment. So here's a good example of that. When you look at this, you likely see a cube. And the reason that you see a cube here is kind of through Gestalt psychologies. These Gestalts, how we perceive a whole. We perceive this whole as a cube, even though what we really have is a series of 8 circles that have lines drawn out of them. Okay, so really what we have sensation-wise is very different than what we're perceiving as the whole. So this whole we're probably seeing is the cube, even though there really are these 8 circles that are kind of giving us the impression of a cube that may or may not be there. So again, this is that idea of Gestalt psychology, that the form, the whole, is greater than the sum of its parts. So these 8 circles don't mean that much, but what our brain perceives is a lot more than that. Our brain perceives a lot more than just those 8 circles. Our brain perceives this whole cube, again, because of the way Gestalt psychology works, because our brain views the whole as more than the sum of its parts. Okay, so here's some other examples of this, how this works. So again, this part of seeing this Gestalt, or this whole, we can see faces and other objects as distinct from their surroundings. So this is, again, about seeing the thing, the form, the figure, okay? So how do you decide that your device is separate from everything else? How do you decide that that cube is there, that's separate from those 8 circles? How do you decide this stuff? And one of the ways Gestalt psychologists study this is what we call figure-to-ground illustrations. So you've probably seen this before. You can look at it 2 ways. This is what we call an ambiguous image, because you can either see 2 faces looking at each other, or you can see a vase. And that depends on whether you're looking at the blue as the figure and the white as the background, or if you're looking as the white as the figure on a blue background. So you can see it one of 2 ways, depending on what you look at as the figure, okay? So when they say figure to ground, they're really talking figure to background, okay? So don't get confused what we're talking about. We're really talking figure to kind of background. So a figure to ground is, how do you tell that the clouds are the figure and the blue sky is the background, right? That your device is the figure and everything else is the background. That's what we're talking about here. And these figure-to-ground illustrations, these ambiguous illustrations really are a nice way of seeing, again, that that's just this gestalt, that the whole is greater than the sum of its parts. Because this can be viewed two different ways. This can be perceived two different ways, even though the input, the sensation is the same. That image doesn't change, whether you're looking at it as a vase or whether you're looking at two faces. So that sensation doesn't change. That visual information that's coming into your brain now through the photoreceptors is not changing. What is changing is that top-down, that perception of it. So again, figure to ground is one way Gestalts talk about organizing the visual field into objects that stand out from their surroundings. Another way that they talk about doing this is through grouping. And this is how we organize stimuli in our visual field and what we're seeing into meaningful groups. And we'll talk about 3 different kinds of ways to do that. Your book talks about 3. There's more than this, and so I'll kind of allude to some others here in a second. But the 3 that we're really going to focus on, and your book does a good job of that, is proximity, continuity, and closure. So proximity, so grouping is, again, how we organize these stimuli into some kind of groups. Proximity is an example of here. So really what we have here is 6 lines, okay? What you probably perceive is 3 columns, or maybe 2 columns, depending on how you're looking at it. And the reason that we do this is because of the grouping. Because of the spacing of them, we see that the far 2 left are a little bit closer together than the middle 2, and then the far right 2. And so we're going to perceive those as 3 wholes instead of 6 separate lines. And so this is grouping by proximity, how close something is together. Another one is similarity. You're more likely to see this as 3 columns, 2 of triangles and 1 of circles, than you are to see it as 3 rows because of similarity. You're going to group the triangles together and you're going to group the circles together because they're more similar. Another example of this is continuity. Continuity, we'll see here you're more likely to see a straight line with a squiggle line through it than you are to see a series of half circles. Okay, because again, your brain wants to organize things by continuity. These are all ways that your brain wants to perceive things. This is that top-down processing. So again, you're more likely to see these a squiggle line with a straight line through it than you are to see a series of half circles just kind of butted up against each other. Another one that we use is connectedness. You're more likely to see 3 barbells here than you are to see 6 dots and 3 lines because there's this connectedness between them. And the last one is closure. Closure was what was being used in that first example of the cube. It's an illusory contour because we didn't have closure in that one, in those blue circles. And because we don't have closure in the left image here, you're more likely to see something with air quotes than you would in the right image. Because we don't have that closure in the left image, we can see a triangle that's kind of placed on top of those circles. Where in the right image, we don't have that because what we have is the closure of those C's. So again, closure is another cue that we use to either group something or not. Okay, so that's kind of our brief overview on shape perception through Gestalt psychology. The next one we're going to talk about is depth. So really what you have in terms of sensation is you have 2D information. You have information about height and width. You don't have any information about depth, okay? Your sensation-wise, nothing's coming in. But the brain is really, really good at taking that 2D information and making it 3D. And it does it through a number of ways. And again, in a perception class, you would spend entire lectures just talking about depth perception. Again, multiple. We can have different ways to do this. We can have monocular cues, which are just cues that you can see with just one eye. We can have binocular cues, which are the ones that we're going to use with 2 eyes. And again, this binocular vision is really one of those benefits that we have. Depth perception is really one of those benefits that we have because of binocular vision. So again, this is allowing people to gauge distance. The other piece of this that's really helpful is our world experiences. So our world experiences really will shape this ability to develop depth perception. If the right input is given, this will develop properly. If the right input's not given, depth perception cannot develop properly. So one of the ways that we test the development of depth perception is the visual cliff. This video that you're being asked to watch, it's linked in iCollege, shows you the visual cliff. It talks about what babies typically do for the visual cliff. So basically what it is it's a fake cliff. There's a plexiglass drop-off. Infants can crawl across the plexiglass, but underneath, they can see the pattern goes away, and if they have depth perception, often they won't do it because it looks like a cliff, even though it's not. It's a visual cliff, even though it's not a real cliff. And what we'll see is new crawlers don't want to crawl. New crawlers will crawl across it no problem because they don't have that depth perception. What we see is that self-locomotion, being able to move yourself around, really promotes depth perception. And so this video, what they did is it's a video of a study where they introduced pre-crawlers to self-locomotion. They gave them a little car they could drive, and so they could learn about moving themselves around. And what they saw is once they were able to crawl, they did not want to cross the visual cliff, just like somebody who'd been crawling for a lot longer. So again, it really is this self-locomotion that's really promoting depth perception. So again, make sure you watch this video. There will be questions on this on the assessment. The next one we're going into is motion. So humans really have specialized motion detectors. Again, our rods really help us with that. They're really good at motion on our periphery and those sorts of things. But we can also have things called apparent motion. So if you're looking at this image on here, it kind of looks like it's moving a little bit in the center. And if you move your head around it, it totally looks like it's moving. And the reason that it's doing that is because even though these visual stimuli are static, they're really appearing at two different locations at the two different times. And so your brain interprets it as motion. Now, this image is a little bit harder to understand what you mean by apparent motion as opposed to the flip book that we had as children. And so apparent motion is that flip book technology, right? And I say technology in a loose term, though I'm sure it was invented at some point. And it's, again, these visual stimuli are presented at different locations in a sequential manner. You're like, what are you talking about? This is what I'm talking about. So again, here we have, it looks like Michael Jackson's dancing, because even though we have different images, they're being presented at different locations in a sequential manner, and that creates apparent motion. This is, again, how cartoons were first started when they were drawn by hand, is how this started. So again, apparent motion is really these visual stimuli are presented at different locations, but in a sequential manner, which gives us this perception of motion, even though there isn't necessarily motion there. Another thing that we do that we can use motion for is that figure to ground organization. So again, figuring out the figure versus the background. In this situation, there's a fish here. Can you see the fish? Probably not, because this fish is really, really well camouflaged. But if we add motion into this, we'll be able to see the boundaries or the edges of this fish that's otherwise really well camouflaged. So once he swims away, starts swimming around, like, oh, hey, look, there's a fish there. But he blends in so well that you otherwise have trouble seeing that. And so this motion can also be a cue for figure to ground organization. I mean, he's really well camouflaged. Even when you get close, you're like, and he stops moving, you're like, do I see him? And then he starts moving, you're like, yep, there he is. The last one that we're going to talk about is constancy, these perceptual constancies. And perceptual constancies is this idea that our brain, through, again, top-down processing, recognizes that objects don't physically change just because the sensation information that's coming in changes. So this is that your red shirt is red, whether when you put it on in the bright daylight in the morning and when you go to bed at night and it's dark. Even though the input that your brain is getting on your red shirt changes, that light refracting off of it changes, you don't perceive the red as changing because, again, of perceptual constancies. This is the fact that when you open a door, you don't think the door changes shape just because you're seeing the edge of the door versus the front of the door. This is this idea that objects have consistent color, brightness, shape, and size, regardless of that sensory information, that retinal image that we're getting. So if you have something, you see a friend down the hallway, you don't think they're shorter because they're down the hallway than when they're close up. You just think they're further away. You use that as a depth cue. That perceptual constancy of height allows you, of size, allows you to use that as a depth cue. So the three that we're going to talk about are size constancy. And this is, again, that perception that objects have a constant size even though the distance from them varies, okay? So in this image, you're going to perceive those columns those blue columns all as the same size, and that just the last one is further away, not that it's actually smaller. So that's, again, that same thing. You see your friend in a hallway. You see a dog down the street. You don't think that dog is a Chihuahua instead of a German Shepherd because it's far away. You just think it's far away because of size constancy. Another one is shape constancy. This is that idea that, again, just because you open a door and the actual shape of that image changes from a straight rectangle to a trapezoid doesn't mean that you think the actual door changed shape, right? When you look at somebody from the side versus face on, you don't think that their head changed shape just because you're getting a different information on your retina. These are those perceptual constancies. And this last one, color constancy, is the same way. But some of the internet kind of things that we've seen really will play on those. So the dress, whether you saw it as white and gold or blue or black, this is really just playing on your color constancy. So you use background cues and lighting cues about color constants to maintain that color constancy. And those were manipulated in this situation so that you could either determine it as blue and black or white and gold. But this was an illusion that was created out of your brain's willingness to have color constancy. That, again, your red shirt is red, whether it's red at 7 o'clock in the daylight or it's, red at 11 p.m. in the darkness. You still see that as red even though those wavelengths change. This is that even if you're reading, in a dark room versus bright outside, that looks like black ink on white paper. It doesn't, even though you might be getting different visual information, sensation information from the actual wavelengths that are hitting your retina, because of color constancy, you're going to perceive that as the same. Okay, this ends our conversation about vision. Thanks so much.